Our first speaker this afternoon is such a transplant. Stephen Minger is from King's College in London, where he's a senior lecturer and director of the stem cell biology program. He first pursued research in neural stem cell biology as a postdoc at UC San Diego. After additional postdoc work at the University of Kentucky, he was appointed an assistant professor in neurology at that medical school in 1995. In 1996, he moved his research program to Guy's Hospital and was appointed lecturer in biomedical sciences at King's College, London in 1998. In 2002, Dr. Minger was awarded one of the first 2 licenses granted by the Human Fertilization and Embryology Authority for the derivation of human embryonic stem cells, and his group generated the first human embryonic stem cell line in the United Kingdom. Dr. Minger's laboratory is primarily interested in the generation of enriched populations of tissue-specific cell types from pluripotent embryonic stem cells for the use in cellular replacement strategies for treating human disease. Today he's going to review embryonic stem cell research going on around the world. Steve, thank you for being here. Let us welcome Dr. Minger.
Thank you very much. So I'm very pleased to be here yet again, and I'm grateful to Professor Lane, Professor Shine for the invitation, and to May as well. So what I'll do is I'll take you through what our lab's been doing, probably provide a little bit of additional detail to complement Mark's talk about the situation in the UK, and then tell you where we're planning to go, which is basically around therapy. So my lab and me basically got interested in stem cells quite a long time ago, and it's really pioneering work like this that really was the impetus for this. So this came from Anders Bjorklund and Oli Lindvall's lab in Lund. This is human transplantation for Parkinson's disease. So here's a gentleman, prior to transplantation, about 60% of his time is spent profoundly Parkinsonian, even though he's on anti-Parkinsonian medications, transplanted with human fetal tissue, and slowly over about 4 years his symptoms resolve. He's now about 16 years post-transplantation and remains off of Parkinsonian medication and free of symptoms, at least on his worst affected side. What these graphs do is provide dopamine to the brain. You can see that using positron emission tomography. After about 4 years post-transplantation, he's back up into the normal range of dopamine metabolism, and that's actually increased over time. These graphs don't cure Parkinson's disease. It's one thing that I get a little annoyed with when we talk about stem cell therapy is the idea of cure. We don't cure anyone. We alleviate symptoms, but the underlying disease is still there. And it's exemplified in this guy because he only was transplanted on one side, contralateral to his worst affected side, and you can see what's happened. His brain has continued to degenerate. But these graphs not only are stable and provide long-term therapeutic benefit, they actually improve over time. So as his brain is degenerating, his graphs are actually getting better and better at working. Now the problem with this is, as many of you will know, that this relies on aborted human tissue. This gentleman was transplanted with 5 embryos of about 7 weeks gestational age, and currently the Lund group would want to transplant probably 12 to 14 individual human embryos into a single patient. So we have a cell-based therapy that's clinically very effective, but because of a profound shortage of tissue, this will never be more than really an experiment. The Lund group has only transplanted 20 patients in 18 patients in 20 years for this particular reason. This, like islet transplantation using the Edmonton protocol, both are clinically very effective cell therapies. But again, because of the profound shortage of tissue, these will only be used in a handful of people. So what stem cell therapy is designed to do is provide the missing cells. And that's really what my lab is predominantly focused on. So just so we're all playing from the same book, stem cells are basically cells that are capable of unlimited self-renewal. That is, every time a stem cell divides, it gives rise to a daughter stem cell. That's what stem cells are. They're a cell population that never runs down. They have unlimited self-renewal. They're multipotent stem cells, which are generally defined as being tissue-specific. So liver stem cells give rise to all the cells in the liver, brain cells, stem cells to cells in the brain. Or there are some unique pluripotent stem cells that are capable of giving rise to all cell types in the body. My lab has worked on stem cells from a variety of different tissues, so I'll show you in a moment. I would argue that at the moment, the only pluripotent stem cell population that we are aware of that's been very well characterized is embryonic stem cells. The others, I would argue, are multipotent. So stem cells are most readily attained from developing tissues during fetal development. And my lab has made a cottage industry, really, of going into tissues and isolating stem cells from various tissues, as I'll show you in a moment. We also more recently have begun to work on what are called adult stem cells, but in this case, not stem cells from bone marrow, but rather adult neural stem cells in human brain, where we have evidence of some regenerative capability that's under review at the moment. My good friend Colin McGuckin will talk about his cord blood stem cell work, so I'll ignore that for the moment. I'll spend quite a bit of time talking about embryonic stem cells. Now, when I went to Fred Gage's lab in 1992, we knew that cell therapy for Parkinson's disease would work, but it was never going to work if we had to rely on fetal individual embryos for transplantation. So when I went to Gage's lab, the idea was, could we take cells from developing brain and expand them to the equivalent of 12 or 14 embryos? Could we have an unlimited source of cells for transplantation? And when we began to think about using stem cells for therapy, we came up with this shopping list. Now this seems really simple. There's no rocket science in this. Stem cells must be able to proliferate for very long periods of time, and they must generate huge numbers of cells. Let me give you an example, so put this in some context. To transplant somebody with islets and get them off of insulin requires about 1 million islets. Each islet will contain 2,000 to 5,000 cells. So per patient, you need to generate about 5 billion cells. If you want to transplant 10 patients a week, that's 50 billion cells a week that you have to produce. And that's just for type 1 diabetes. If you also want to do this for Parkinson's disease and for other diseases, you're talking about having to generate trillions, gazillions of cells. And you're starting with a very small population. In particular, if you're starting with adult stem cells, they don't have the ability currently to expand to those kinds of numbers. Or if they do, there's concerns about how stable they really are. So in any stem cell population we're thinking of, the self-renewal capability is crucial. And the cells have to be stable over time in culture. They can't change their phenotype during this large expansion phase. And then they have to be able to be transplanted. They have to differentiate into the appropriate cell types. They have to integrate generally into the host tissue. We'll talk about the immune response in a moment. And they have to provide long-term sustained therapeutic benefit. So this, again, is really simple, but this for us is the mantra of whether or not an individual stem cell population has true therapeutic potential. So starting in '92, we began to go into developing brain and generate neural stem cells. And we still continue to do this, although, as I'll show you in a moment, it really has very little therapeutic value. But we can grow these cells fairly easily on a monolayer in defined culture conditions, and they differentiate into the appropriate cell types in the nervous system. This gives you an idea of their expandability. So this is in the millions over here, days in culture here, using this cocktail of FGF2 and some conditioned medium from a human tumor line. In 50 days, we can go from 1,000 cells to 500 billion cells. Okay? So these cells are highly expandable. Okay? And over the years, we've generated a wide variety of different stem cell populations from brain. We've shown that we can transplant these into a wide variety of lesion models. We get behavioral recovery in some, but not all, of the models that we've looked at. We can genetically engineer these cells, and we get long-term sustained gene expression after transplantation. But the bottom line with this is, despite the fact that we have worked on these cells for almost 15 years now, no one in the world has established a neural stem cell population from rodent or from human brain that has any therapeutic value whatsoever. Because despite the fact that we can expand these cells to very, very large numbers, the cells that are required for therapeutic efficacy, that is, cells that make dopamine, cells that make acetylcholine, motor neurons from the spinal cord, those cells are gone after about 3 weeks of expansion. The other problem with this is, as the cells expand in number, you get fewer and fewer and fewer neurons. You get more and more and more astrocytes. And astrocytes are not therapeutically of really any benefit at all. 15 years worth of work, probably 300-400 labs, and nobody has developed a stem cell population with true therapeutic value. Okay, so it's been pretty much a waste of time. Now, not all stem cell populations from developing tissues have these kinds of limitations. We've established these stem cells from developing retina, which are quite interesting. And probably more importantly, we've established, both from human as well as from rodents, clonal populations of hepatocytes, which have been in culture for over a year now. And these cells remain functionally active. They don't change phenotype. Once established, they're very stable. So you have to look at stem cell populations on their own merits, compare one against another. These, I think, will be quite important, both for therapy as well as for drug testing, something we've talked about using stem cells for already. Embryonic stem cells, though, in my mind, probably offer the most potential. These cells are obtained from the developing blastocyst from the inner cell mass. And what we do is we take these cells out of a blastocyst at a time prior to them beginning to see signals which begin this specification process. That is, when these uncommitted, pluripotent, undifferentiated cells begin this process of differentiation into the three main germ layers and ultimately into all the cell types of the body. This is a trick, though. This is a trick of tissue culture. We take these cells out before they begin this process and we trick them. We keep them in this state, we block this differentiation step, and they expand in number. And if you do this right, and if you're careful, you can keep them in this state literally forever. Once established, these cells are immortal. They don't age over time like somatic stem cells do. Their telomeres don't shorten. And if you're careful about how you grow the cells, they remain genetically normal. Okay. So, because of the problems I alluded to with the neural stem cell work, about 8 years or so ago, we moved into mouse embryonic stem cells. And we spent a long time playing around with them. And actually, mouse embryonic stem cells are quite easy to work with. We had a very good cell line, this one here, E14-2. It grows in the absence of feeder cells, and it grows in lymph and serum, and it's highly expandable. And we got very good at differentiating it into a wide variety of tissue types. So we could generate lots of neural stem cells that made neurons. We could generate muscle, retinal stem cells, and a whole variety of other tissue sources. So that was all very fine. And then one day in January 2001, the laws changed in the UK, and Mark, I know, has already talked about this, where House of Lords and House of Commons allowed for extension of the Human Fertilization and Embryology Act. So we had been trying to get access to the first stem cell lines that had been made by Jamie Thompson and the group in Singapore. And in 2001, we were told that it would probably be 2 to 3 years before we would get cells, simply because the waiting list was so long and there were so few cell lines. So, excuse me, when the laws changed in Britain, I thought, well, maybe we should try to do this. So I approached Professor Peter Brodie, who heads the Assisted Conception Unit at Guy's at King's College London, and he and I and Susan Pickering agreed that this would be something worth pursuing. So we put in a license application after we got local ethical approval, and we were granted one of the first 2 licenses together with Austin Smith's group in May 2002, and it's just now been renewed for 3 additional years. Our license is very succinct, and it's very clear about what we can and cannot do. We're allowed to derive human embryonic stem cell lines from embryos which have been created by IVF and have been donated under informed consent. In our hands, the patients are offered no financial or medical inducement whatsoever, and basically they sign away all rights to anything that comes out of the stem cell lines. So if the lines are used for commercial purposes and somebody makes money off of it, the patients get nothing. They literally donate freely for goodwill. Our license does not allow us to create embryos for stem cell derivation, but we could do that. We could approach the HFEA for the ability to do that, but we haven't yet. All cell lines by my group and any other group in the UK have to be deposited in the UK Stem Cell Bank. The bank will make those cell lines freely available to anyone who wants them. They are currently available now, and they are currently free. So unlike the $5,000 for $6,000 that Geron, YCell, ESI charge, all the cell lines in the stem cell bank are free. So I spend 6 months making a stem cell line. I put it in the bank, and the bank then passes it on directly to my competitors, and there's nothing I can do about that. But I still think it's actually a very, very good thing, and I'm very supportive of the bank. They've been really very, very, very supportive and are good collaborators. Somatic cell nuclear transfer in the UK is legal and under license. There are 2 groups who have licenses, the Newcastle group and Chris Shaw at King's together with Ian Wilmot at Roslin. I'll talk a little bit more about this at the end. And unlike the US, cell nuclear replacement for reproductive purposes is completely banned in the UK. One point that I want to make, and this is apropos to Korea, is before anybody did any work in the UK, this was all in place. So after the House of Lords had voted to allow this to happen, we had a year-long consultation process with a select committee. And basically, they went through and they looked at every aspect of this before anybody started the work. It was only after the committee's report had been handed in and it was clear what was going to be allowed and not that we were then granted licenses. So the UK has been very pragmatic and I think very prescient in how they've set this up. And also by taking stem cells and putting it onto the back of existing regulations. Unlike the US, the UK, all reproductive medicine in the UK is very, very tightly regulated by the HFEA. So putting stem cells on the back end of already pre-existing legislation is also quite a smart thing. So how do you make human embryonic stem cells? You need a source of human embryos. This is in vitro fertilization, all done in a dish. So this is immediately after fertilization. You see the two individual nuclei, they've not yet fused. After fertilization, you grow the embryos in culture. And in our clinic on day 3, 2 best embryos, and only 2, are implanted. Any other high quality embryos like this one, we would encourage the patients to freeze. So if the first implantation fails, we don't have to go back, stimulate, and retrieve eggs again. Any other poor quality embryos at this point are generally discarded. They are deemed not to be suitable for implantation and would probably not give rise to a viable pregnancy, so they are discarded. And this is the standard practice in IVF clinics, at least in the UK, and I'm certainly true in the US as well. What that means is, in our clinic, if the best embryos go to the patients and the worst ones are already on their way out, what do you have to make stem cells from? Most groups would only have access to these embryos here, which are not suitable for implantation. But currently, to generate human embryonic stem cell lines, you have to culture the embryos for 3 additional days to get to this state. And if they're already on their way out here, they're not going to progress. But that's generally what most groups have to work with. The only exception to that is if you have a very poor freezing program, and many clinics do. So you will implant 2, and then anything that doesn't get implanted could be made available for stem cell research. We have problems with that, personally. We don't like that at all. So we don't use routine IVF embryos for generating stem cells. Predominantly what we use are embryos which come from our pre-implantation genetic diagnosis program. So for those of you who don't know what this is when couples have a known genetic disorder, or they're at risk of having a child with a genetic disorder, and they want to try to avoid that. So they come in, they undergo IVF, and at day 3, we remove a single blastomere from each of the embryos, and in many cases we do single-cell PCR to definitively diagnose affected embryos from unaffected. And obviously affected embryos will be generally discarded because they have or are at high risk of having a genetic disease. So we would only use these embryos. Now, like everything related to reproductive medicine in the UK, PGD, or pre-implantation genetic diagnosis, is highly regulated. Our clinic is the second most active in Europe and by far the most active in the UK. And these are individual specific diseases for which we have a specific license from the HFEA to diagnose embryos. Now, all of these translocations basically cause recurrent miscarriage, and we've not used any of these at all. Each of these disorders, we test individually each embryo for these. So if a family comes in that's at risk of having Huntington's disease, we will diagnose each embryo for Huntington's disease, or similarly for cystic fibrosis. So these embryos will all be affected. The X-linked disorders, though, are much more complex genetically, and we don't have good genetic tests for these disorders at the moment. But these only affect males. At worst, females can only be carriers. So what we do here is we sex the embryos, and all the males are generally discarded, and only females are implanted. So what that does for us is it gives us access to a pool of embryos, some of which will be genetically normal, but which are of very high quality because they come from fertile patients. These embryos, on the other hand, will all be affected. Now you might say, what's the point of creating a stem cell line that's got cystic fibrosis, for instance? Well, that's exactly the reason why we want to do it. We want to create disease-specific stem cell lines. Stem cell lines that encode these important genetic mutations and use them as a tool for fundamental biology, for drug screening, for toxicology, for gene targeting, and for developing new therapies. So we're very interested in this, and we're trying very hard at the moment to generate cell lines against spinal muscular atrophy and Huntington's disease. To give you some idea of how complicated this is the size of us, basically, 3 days after conception. This is the sharp end of a sewing needle. Okay? And what my colleague Susan Pickering does, I can't show you the movie, unfortunately, because it won't run on anything other than my computer, is she very carefully removes a single blastomere. This embryo usually has a membrane around it called the zona pellucida, and she makes a small hole in it. She very carefully removes a single cell and then does single cell PCR and tests that. If the embryos are donated to our research program, we culture them until day 6 generally, when the blastocyst hatches out of the zona pellucida you see here. So this is only about twice the size of what I just showed you. This small little population of cells here, the inner cell mass, this is what turns into a human being. This larger structure, the trophectoderm, turns into the placenta. So what Susan and I discovered very early on was if you take the entire blastocyst and put into culture, we never could get stem cells. So what we had to figure out how to do, was basically isolate the inner cell mass away from the trophectoderm. Now, when we started this in 2002, there were about 5 groups in the world who had successfully made human embryonic stem cell lines. So there was nobody that we could call up and sort of, how do you do this? What works, what doesn't work? Now there are quite a large number of groups in the world who have done this, and we've been instrumental in helping 2 other groups establish human embryonic stem cell lines. But with trial and error and with a lot of luck, we were successful. So about a year after we started, we made our first stem cell lines, actually our 3rd stem cell line. We lost one to contamination and another one that arrested after about 50 days. But this one, WT3, was the first one generated in the UK. It looks like an average human embryonic stem cell line. And then we've gone on over the last couple of years to generate 2 additional stem cell lines. So WT3 and this one, WT4, are both genetically normal. WT4 came from an embryo that was at high risk for having muscular dystrophy, but turned out to be genetically normal. This cell line, though, is not. It came from the only embryo that we've ever had who had what's called the delta 508 mutation, which accounts for about 70% of humans with CF. So this embryo was homozygous for the delta 508 mutation. As far as I know, this is the only human embryonic stem cell line in the world that has this CF mutation in it. And we've had about 10 requests from groups who work on CF around the world, mostly gene targeting people, to gain access to this cell line. We're sending out the first cells this week. So we've generated 3 human embryonic stem cell lines, 2 of which are genetically normal. One has this delta 508 mutation. All of our stem cell lines meet the standard criteria of human ES cells. They express all the relevant stem cell markers. They express high levels of OCT4. They express all the relevant stem cell genes based on real-time PCR. And unlike a number of other groups which have found that their stem cell lines become genetically unstable and a bit deranged over long-term culture, we've karyotyped our cells out to passage 45, and they remain genetically normal. But this is probably because we grow very small numbers of cells and we physically dissociate them into small clumps and physically move them. We haven't tried to grow them on a large scale with lots of trypsin. And they're pluripotent. They turn into cells of all 3 germ layers, so we can make liver cells, muscle, neurons, and a few other things. The cells, like all human embryonic stem cell lines, are capable of being differentiated into a wide variety of cell types. This will give you some idea of how difficult this is. This slide's about not quite 2 years old, and we have only had about 170 embryos in 4 years. And that's about less than 5% of probably what most of the other groups I know have had, because they're reliant on really everything that comes through fresh IVF, and we haven't taken that route. Up until very recently, it had been a year since we'd had any embryos, so we haven't had very many. But these are the best embryos that you're going to get, because these guys here all come from fertile patients. These cryopreserved embryos, we have a very good freezing program at King's. Our pregnancy rate from frozen embryos is almost the same as it is from fresh. So we freeze high quality embryos very well. But of the 100 embryos that we've had, you can see only about half actually progressed from day 3 to day 6. Of those, only about 2/3 actually had an inner cell mass, despite the fact that they were blastocysts. Of that, only half actually, when put into culture, did anything. And of the ones that did grow out, only 20% actually generated stem cell lines. So this efficiency of 5% is actually very good. Most other groups I know have had thousands of embryos and have made 1 or 2 cell lines. So even with the best quality embryos, this efficiency is still very, very low. And I think this is important when we begin to talk about somatic cell nuclear transfer, because the efficiency of that is going to be even less. So now that we've made stem cell lines, what do we do with them? Well, my lab has always been interested in the therapeutic applications of stem cells. So rather than trying to grow large numbers of embryonic stem cells, which are bloody difficult, these cells are really hard to grow. And you have to be very, very good to keep them undifferentiated, free from mycoplasm, and expanding. Our goal is to actually drop them down a step or two and generate these somatic stem cell populations, which are much easier to grow. Oftentimes we can grow them in chemically defined media with individual growth factors. And once these are established, they're very easy to maintain. And we can grow these cells into the billions. This cell population, I'm not convinced you can grow into the billions and direct its differentiation appropriately. So this is our goal: establish these somatic stem cell populations and get them into preclinical models. So this may look like a wish list or a big shopping list of potential therapies, but these are all the things that my lab currently works on, and these are all funded projects. So for CNS disease, we've had a long-standing collaboration with Steve McMahon's group at King's. Steve is one of the world's leading pioneers in spinal cord damage, and particularly neuropathic pain. So together with Liz Bradbury, who's a lecturer in his group, we've spent the last 3 years transplanting cells into a wide variety of traumatic injuries to spinal cord, and more recently in animal models of multiple sclerosis. So this work is actually progressing very well. With Susan Duty and Professor Peter Jenner at King's, we have work trying to differentiate human embryonic stem cells toward a dopaminergic phenotype, and this new very large bit of work on adult neurogenesis with Clive Ballard. I'm really fortunate to be part of something called the UK Cardiac Cell Therapy Consortium. This is a group of 8 or 9 interventional cardiologists, that is, guys who go into your femoral vein or into your wrist and put things into your heart without cracking your chest open anymore. This is put together really by Anthony Mather and John Martin in London, and basically it's to move cell therapy into heart disease. So currently we have 2 funded clinical trials of bone marrow transplantation into heart, one for myocardial infarction and one for chronic heart disease. But together with these interventional cardiologists, there are 10 or 12 basic stem cell labs, mostly working on hematopoietic stem cells and mesenchymal stem cells. But the idea is, we don't think bone marrow is the appropriate source of cells, but that's already in the clinic. We want to devise specific cells, cardiac cells, and together with Karen Hershey, endothelial progenitor cells, to actually put cells that belong in the heart back in the heart. So right now we're using bone marrow because that's what's available, but the long-term goal is to generate cardiac progenitors, cardiac stem cells. And we have another program of work with Professor Robin Ali, who's at Moorfields Eye Hospital in the Institute of Ophthalmology. Robin is a world-class ophthalmologist, if I can spit that out, and he has big research programs in macular degeneration and retinitis pigmentosa, so we're generating retinal stem cells to test in his animal models. And then we've had probably our longest collaboration has been with Peter Jones, Shanta Prasad, Stephanie Amil, and the King's Islet transplant program. So King's has, as far as I'm aware, the only islet transplant program that's active in the UK. We've transplanted 4 patients, one of whom is completely off of insulin. The other 3 are still on some low-dose insulin. But this has really proved quite disappointing because although mouse embryonic stem cells can be differentiated into insulin-producing cells with some efficiency, sort of around 10%, if there is a cell that human embryonic stem cells don't make with any great efficiency, it is a beta cell. We don't really understand this. I know of at least 10 other groups who've also been funded by JDRF over the years, and we get together at meetings and we really sit and scratch our heads because none of us has figured out a way to generate large numbers of beta cells. The only group that I'm aware of who has any real success with this is Alan Coleman's group in Singapore. They have worked with 6 stem cell lines. One of them, they can get about 10% of the cells to differentiate into beta cells, and this is after 5 years of hard work. The other 5 lines, like ours, all make cells with an efficiency of much less than 1%. We don't really understand why this is. Like Parkinson's disease, diabetes represents a very good clinical target because we know how to transplant these cells in man. We know how many cells are required. We know where to put them. We have the immunosuppressive regimes. We know how to make this work, and yet we can't make the cells. So this has proved rather disappointing. I'll show you in a while what sort of our approach to this has turned out to be. It's quite different than what I thought it would be. And then we have some newer programs, one on generating liver cells together with the King's Liver Transplant Program. This is one of the UK's largest liver transplant programs. But there's a real need. A lot of kids in the UK who die awaiting liver transplant. And then programs that are funded on generating cartilage, bone. This is a very interesting program by my good friend, Professor Paul Sharp. This might sound a little bit dubious, but actually Paul has figured out how to turn just about any stem cell population into teeth. He's done it with our neural stem cells, he's done it with hematopoietic stem cells, and he can specify a molar versus an incisor. Okay? It's very good. Now, I've always had, and he's doing this now with our human embryonic stem cells, but I've always been a little bit sort of, I don't know, irreverent about this. And Paul's work was publicized about 1.5 years or 2 years ago. And I guess because we're in London, because we were the first group to get a license and the first group to make cell lines, we've had a lot of media attention over the years. When Paul's work got publicized, my phone rang off the hook for a solid week. More phone calls than I've ever gotten about anything. I had to unplug my phone. It was amazing, and the number of people who have bad dentation is amazing. You just would not believe it. Maybe it's the Brits. I'm not really certain, but this. And I think, for the obvious reasons, I think Paul will be in the clinic long before most of these other things get there, and it seems to be a really big, pressing medical need. And then together with Chris Mason, who's another good friend. Chris is an engineer, basically, and a surgeon, and they have a very, very large bioprocessing, bioengineering group at UCL. And so together with him, we're working on how we scale this up to take cells ultimately to the clinic. But before we do, we've got a few things that we have to solve along the way. Currently, all the cell lines, with rare exceptions, and I mean rare exceptions, worldwide, are not suitable for human use. They have all been generated either in serum or in mouse feeders, or even if they've been raised in synthetic sera, that has animal products in it as well. It's clear in Europe that we will not be able to use cell lines which have been grown under any of those conditions. So one of the things that we have to do is we have to figure out how to grow these cells in chemically defined media with no serum and probably with no feeder cells. So that means we've got to identify the growth factors that the fibroblasts are making that allow these cells to expand in number but block their differentiation at the same time. So we have a big program of work on that. Then there's the issue of controlled differentiation. How do you take a cell that wants to turn into over 200 different cell types and reproducibly and consistently turn it into the cells that you want it to turn into and not everything else? The problem with embryonic stem cells is when they differentiate into a lot of different stuff. And they do it according to what they want to do, not what you want them to do. So this is a really difficult problem, and I'll talk a little bit more about that in a moment. Then we've got to test the cells in animal models. And this is one thing that we're beginning to realize is that a lot of the animal models we have for human disease don't recapitulate human disease. So our spinal cord injured rats, 50 days post-injury, they're back to normal. They recover. We're putting cells into mouse and rat heart, which is physiologically nothing like human heart. And the animal models of myocardial infarction and heart disease are very different in rodents than in humans. So, we may need to do a lot of this work in pigs and in primates. You mentioned primate research in the UK. It's very dangerous. It's like, being an abortion doctor here. It's not a good environment to be doing primate research. But this may be something that we're going to have to really think about. Then we have to think about how we're going to deliver the cells. So it's clear that if we're going to put cells into the brain, a cell suspension is fine. If I put in 10 million cells that are neural stem cells, and 20% of those cells can differentiate into dopaminergic neurons, you can treat people with Parkinson's disease. The problem begins when you look at other therapeutic applications. So for instance, in diabetes, if we were to inject 5 billion cells that make insulin, they would do nothing. Because individual beta cells on their own do not respond to glucose in an appropriate physiological manner, you have to aggregate them into an islet. Now, how we do that, I don't know. My pal Peter Jones claims that he knows how he can make what he calls pseudo-islets. But, you know, it's not just enough to make the cells. Now you've got to configure them into something that's appropriate for treatment. And then for other things, we may have to work with scaffolds. We may have to make three-dimensional tissues. And all of this is new. We don't know how to do a lot of this. Probably the bone guys do a bit, but can you make, you know, the right bone of the right consistency and the right shape? So that's that problem. And then here we go, the immune response. This is when cell biologists stick their head in the sand because we don't know what we're going to do about this. My new vice principal, Robert Leckler, is a transplant immunologist, and I, on a routine basis, get tutorials on immunology. Immunosuppression is what we traditionally use, but if we're talking about young kids who don't have a life-threatening form of diabetes, you're not going to want to immunosuppress them. But you would also like to, you know, give them the benefit of cell therapy. Somatic cell nuclear transfer, well, we don't know if this works in humans. It's clear that Professor Wang did get blastocysts. He got successful nuclear transfer, and he got them to the blastocyst stage. Whether or not there are cell lines that came out of that work, we don't know. There's still an ongoing investigation in Korea. I looked this morning at the newspapers, and somebody who's close to the prosecution has said that he's admitted fabricating the data, which is the first time he's done this. Now, whether or not that's true or not, I don't know. Whether or not this does work remains. Nobody else has been able to replicate his work. Alison Murdoch and my pal Miodrag Stojkovic managed to get something to about the 8-cell stage, and then it degraded. So we still don't know if somatic cell nuclear transfer is possible in humans. I never thought about using it for personalized medicine. I always thought we could create stem cell banks. So you go out and you screen 10,000 people, and you find 1,000 of them that have the widest amount of genetic diversity. And we create a bank of hundreds, if not thousands, of cell lines, and we try to match people to cell lines. The problem there is, given the enormous variability in HLA haplotypes, the number of cell lines that would be required to match people such that you didn't have to use any immunosuppression is astronomical. You would literally need hundreds of thousands of cell lines. So I'm not so sure about this anymore. In some cases, you could probably encapsulate the cells in some kind of polymer that hides them from the immune system. So this would work, for instance, for type 1 diabetes, but would not work in the brain or in the heart, where the cells have to physically integrate into the tissue. Lots of people, including my vice principal, believe that induction of tolerance is the way forward. And this is where you, from a given cell line, you make some hematopoietic stem cells, you infuse them into your potential transplant recipient, you let those cells settle down into their bone marrow and begin to create another immune system. So you now have your own immune system, and you have an immune system that comes from the cell line. And then you come back in at some point in the future with a second transplant. So that sounds good. I like that idea. That sounds very promising. It depends on who you talk to. I talked to some transplant immunologists who say this works really well in mice and it's never worked in humans, and they've been trying to do this for 30 years. My principal thinks it's a great idea, and he's a transplant immunologist, so I don't know. But just, I thought I was in the jungle for a second. So this is a really big issue, and I don't know what we do about this, to be honest with you. I mean, I assume we will immunosuppress in the early days, but this is not something that we are going to want to do, particularly with children. So what all of this requires now, particularly with the new European tissue directives that are coming in, is we will have to generate GMP-like therapeutic quality cell lines. So at King's, we're spending £4 million, which is about $7 million, on creating a GMP level IVF clinic and stem cell derivation suite. So most of these other things I have to have solved by the time this is ready to go. So I need to go back to the lab, obviously. Now, differentiation, this is really a big problem as well. The problem with differentiating embryonic stem cells is that it's difficult to do. And the way most people do it, they form these things called embryoid bodies. So you just take your colonies of ES cells, you break them up a little bit, and you put them into suspension. And they grow these little embryoids. And what these are three-dimensional floating aggregates of cells where the cells begin a differentiation process. Now in mouse, this works really well. They pattern themselves appropriately into mesoderm, ectoderm, and endoderm. Human cells don't really do that. They differentiate, but not in the same way as mouse cells do. The problem is, the good thing is it's very efficient, and you get good differentiation. The downside of that is you get very mixed populations of cells. But if you look at all differentiation protocols, including the one from Geron, which is what they hope to take to the clinic next year, this is what it involves. And this is a very capricious process. The cells are doing what they want to do, not what you want them to do. There's some other ways you can try to make embryoid bodies, but none of these really work very well. What a lot of us are trying to do is co-culture undifferentiated cells with literally a second feeder layer. And the second cell population provides directive signals to the cells to differentiate in a specific way. So let me give you some examples. There's a group in Japan who uses a mouse stromal cell line called PA6, and in their hands, their human ES cells tend to differentiate toward a dopaminergic phenotype. Christian Mummery's group in Belgium uses a similar cell line, but of an endodermal origin, and her cells differentiate, about 70% of them differentiate into cardiac myocytes. The problem is many of these cell lines are of animal origin themselves. They require serum for growth. And in many cases, you have to physically grow the cells directly on top of the second cell layer. So I don't think that this is a very good way to go unless we can use these cell lines to find the growth factors that they are secreting that drive this differentiation. So our approach to this is just to show you that sometimes, we've come up with this protocol, or Hannah Taylor and my group's come up with this protocol, where we only grow embryoid bodies for a couple of days, and in our hands, with the right growth factors, we get large numbers of neural stem cells to come out of this. So this is very recent work. She's managed to replicate this quite a few times now, so we think we're onto something. But more often, what we want to do is we want to be able to specify the cells ahead of time. And there are really two ways of doing this. One is to find some cell surface antigens, some markers on cells, where in a mixed population of cells, you can pull your cell population out that you are interested in. Problem with that is, with the exception of the hematopoietic system, we don't have really good cell surface markers that are unique to most of the stem cell populations that I've talked about. So for pancreatic cells, for hepatic cells, for neural stem cells, we don't have markers. So that's one thing that we're working on. The second way of doing this is to use genetic modification of the cells, to use gene transfer to force the cells to turn into the cells that you want. And what this requires is you need to know transcription factors that get turned on very early in the development of the tissue that you're interested in. And this is why stem cell biology has really married up with developmental biology. It's very important that those two areas of research are working together. So this gives you an idea of what I'm talking about. So early in the specification of the pancreas, once it is basically received antagonism of sonic hedgehog, it begins this process of differentiating into all the cell types in the pancreas. And one of the first things that happens is a gene called PDX1 gets turned on. We know how important PDX1 is because when we put it into our liver cells, they start making insulin. So PDX on its own is necessary and sufficient to begin the whole biosynthetic pathway of insulin. So what we propose to do is put PDX1 into our human embryonic stem cells and try to force them down this lineage. Similarly, in the midbrain, where the dopaminergic neurons are born, there's a whole cascade of transcription factors that get turned on in dopaminergic specification. So Ron Mackay's group at NIH has shown if you put Neur1 into mouse embryonic stem cells, you can get dopaminergic neurons, quite large numbers of them. Austin Smith's group, or Ming Li and Austin's group, has shown that PTX3 is also capable of doing that. But we want to generate dopaminergic progenitor cells. We want to grow large numbers of these. So we're looking at genes that are much earlier in this process while the cells are still progenitor cells. So we're focusing on LMX1B. And the way we're doing this is we've created this lentivirus. It's actually a commercially available one from Invitrogen, but we've extensively modified it. We've created a multi-cloning site where we can literally put any gene of interest in here, and the cells will all be GFP labeled as well. We can select for the cells that we want using antibiotic resistance. So we've cloned the first, 12 genes as of yesterday, and we're currently putting these into lentiviruses and starting to transduce cells at the moment. So the idea is to generate 12 or 14 or 20 different populations of cells, and out of some of those, we hope we will establish very early progenitor populations. Now, I don't think we're going to be able to take those cells to therapy. Not only will they be embryonic stem cells, but they will also have a lentivirus in them. For those of you who don't know about lentiviruses, it's derived from HIV. So I can't imagine the regulators thinking this is a good thing, although RAC has just approved the first trial using lentis in bone marrow cells, so the regulations may be changing. But ideally what we do, if we can establish these cell populations, we would use them to go data mining and try to find cell surface markers on these cells that we could then go back to the native population and not have to rely on genetic modification. Somebody brought up something earlier that I thought was quite interesting, so I'm going to be very risky and talk about this. Somatic cell nuclear transfer is something that we have decided over about the last 3 to 5 months that we need to start doing. One of the things that we would like to do is establish human embryonic stem cell lines that encode Alzheimer's disease mutations. Currently, the HFEA does not allow for PGD for Alzheimer's disease because it's a late onset disorder. So our chances of actually using PGD to generate ES cells that have Alzheimer's disease mutations in them will probably not be approved. So another way of doing this is to do somatic cell nuclear transfer, take individuals who carry those mutations, put them into an egg, and generate a stem cell line. One of the problems, though, is many of us don't like the idea of relying on very, very large numbers of donated oocytes. This is it is a risky procedure. It is unpleasant. And we don't know what the efficiency of somatic cell nuclear transfer is in humans. So our argument, and this is an argument that's shared by Chris Shaw from King's as well as some other colleagues of mine, is we probably shouldn't be doing this using human eggs. We should do this with non-human eggs. So we will approach the government and ask to be able to do somatic cell nuclear transfer using non-human eggs and human cells. And then the issue is, if you do that, what do you have? If I use a rabbit egg for somatic cell nuclear transfer, put a human nucleus into it, and generate a blastocyst, is it a human blastocyst? Is it a rabbit blastocyst? Or is it a bunny man, as some of my friends have termed it? It's an interesting argument because the HFEA regulates embryos. If it's not a human embryo, it falls outside the act. Okay? The other way to do this is, as somebody pointed out earlier today, which is to make oocytes from human embryonic stem cells. This has been done in mouse. It's only been done in mouse. Whether or not these are functional or not remains to be determined. I think they have been tried and they don't fertilize, but this is early days with this yet. And there are people in the UK trying to generate oocytes and sperm from human embryonic stem cells. This is not research that is banned in any way in the UK. So this may represent an alternative. And then the question is, should these be used in fertility treatment if they were functional? So if they were, then, who's your mother? Is your mother a stem cell line? Is your mother who you were implanted into? It raises interesting issues. In the UK, we've said, even if somebody is capable of doing this, these cells wouldn't be used for fertility treatment. But currently, there is no prohibition against using them potentially for somatic cell nuclear transfer. So, being an American in the UK represents an interesting environment, I have to say. First of all, it's a fantastic place to live. But secondly, I think it really is the best place in the world to do this research. So we have really world-class academic research centers. We have people like Austin Smith, Roger Peterson, who's also an American, Peter Andrews, and others. We have the tightest regulation, I would argue, in the world related to human embryonic stem cell research, but contradictorily, we have the most permissive environment scientifically. The government has been, as Mark pointed out, has been very supportive of this research. They gave us about 45 million pounds a couple of years ago. That may not sound like very much compared to California, but that 45 million pounds, or about $70 million, currently funds about 100 research labs, 150 PhD studentships specifically in stem cell research, 3 major stem cell centers, a number of career development awards, and the stem cell bank. So that's money that's been out in the scientific community for 2 years now. And we have the 10-year strategy document that Mark talked about from the Chancellor's office. This number was one number that was bandied around. It was somewhere between 100 million and, 5 billion. We don't know what this number is going to be. But the government has been incredibly supportive of this research, unlike the situation here. We have the UK government-funded stem cell bank, which currently is taking in embryonic stem cell lines but over time, we'll also take in adult and fetal human stem cell lines. It's a GMP facility, and they're really advising everybody else. There are 5 of us now building GMP facilities in the UK, and the bank is being very good about providing resources to all of us for that. We have this unique entity called the UK Human ESL Forum. So this is all the research labs in the UK that currently work on human embryonic stem cells. Most of us are also deriving. And 3 times a year, we get together for an all-day research meeting. So all of our labs go to one site, and we talk openly and freely about what we do. We let our students and our postdocs and our technicians talk as much as they want about what they're doing. There are no restrictions, no confidentiality agreements, no questions of IP. We just talk about the work. And I think this is a tremendous thing. And then Chris Mason and I co-direct, and we're co-organizers of something called the London Regenerative Medicine Network. This is a group of about 2,000 scientists, clinicians, government officials, people from the charities, people from patient advocacy groups. We have a research meeting every month, followed by quite a large amount of wine drinking. That's scientists, you know. But what it does is it's beginning to bring a community together that's beginning to interact. So we have 2 or 3 speakers, and we're having to start to turn people away. We can only accommodate 300 people, and you know, the demand for this is getting so great that now we're having to look for a larger site. But I really think, you know, this really kind of grew out of the Cardiac Cell Therapy Consortium, and I think that's really the way you have to do this. Basic scientists and clinicians have to begin to start working together years before we get to the clinic, not when we're at the clinic. It'll just take too long. So I think this is a really good model for doing that. I just want to close with my pal Wang. So I was fortunate enough to go to his lab about a year and a half ago, not quite, and it really was amazing. There were 10 of us. The UK government, the DTI, continually send out these missions all over the world, and you assess technology in different places. So we went on a 2-week rock and roll tour of Shanghai, Beijing, Singapore, and South Korea, and so this was at Wang's. And what these guys are doing, these are bowls of ovaries. These are cow and pig ovaries. And they get 2 shipments a day from the slaughterhouse. They isolate eggs, and they go into this room here. There's usually 4 people sitting here, 4 over here, 3 guys in the middle. The guys in the middle enucleate an egg about every 30 seconds, pass it on to these guys. They do the somatic cell nuclear transfer. These guys, on average, do 1,500 nuclear transfers a day, 7 days a week, 365 days a year, and they've been doing this for about 10 years. The technology and skill that these guys have is unbelievable. And actually the Korean government now realizes this, and they're starting to think about what are they going to do with the 65 people that were in Wang's lab, and how do they get them back in the lab. So they will put them back to work. I mean, I think all of us who went to Wang's lab, all of us who went there, there's the great man himself. You can tell it's him because he's the only one who looks cool. The rest of us look like, I don't know, bakers or something. But he's got like this Woody Allen sperm suit on. You know, it's really good. I quite like that. But the technology and the skill that these guys had was unbelievable. And so when I saw the papers, I believed them because, you know, particularly after having been to his lab. But I think it shows you, you know, what can happen if you let all of this go to your head. I think in the UK we've been very cautious about overhyping, you know, where we're at. I think we're a long way from the clinic. I think adult stem cell populations like the one that Colin works on, you know, will have more immediate clinical benefit, but I still believe that embryonic stem cells will certainly have their place. And so with that, I'll just acknowledge the people who actually do the work. So these are the guys in my lab. All these other people in bold are PhD students in other labs who've spent considerable amount of time in our group and have contributed greatly to the work. And our research is mostly funded by the Medical Research Council. So thank you very much. I'd be happy to answer any questions.
We're running late, but I think it's important that we take some time if there are some questions for Dr. Minger. Please. Why don't you just identify yourself for him and for us?
I introduce myself?
Yes.
Oh, okay.
Just tell us your name and where you're from.
I'm from Moscow State University, but I work already here 1 year with Dr. Murad in a stem cell facility of Dr. Murad, Nobel Prize winner, and I'm proud to work here. So my question is really about somatic cell nuclear transfer. Whether somebody ever compared the efficiency of normal blastocyte development if we use a nucleus from differentiated cells, comparably if we use nucleus from adult stem cells, for example, bone marrow stem cells.
So the earlier the cell, the more primitive the cell, at least in animal cells, using animal eggs, the efficiency goes up the more primitive the cell. The best cell to use is an embryonic stem cell. You get reprogramming with very high efficiency. The more differentiated the cell and the older the cell, the efficiency seems to be a little bit less.
But with everybody used adult stem cells.
People have used adult stem cells and fibroblasts, and the efficiency is low, as you would expect. It's not as good as with more primitive cells, but it does work.
Another question. Dr. Brady.
Dr. Mingus, you said that you really thought that Dr. Wong's team had achieved this amazing success, and we all did too. I mean, he was in my office, and CNN was there with the camera and everything. How is it that you know that, as you described so well, the difficulty and the slow process it's going to take to get SCNT to work, to think that he could have done it in such a short time?
Well, because he's got a herd of cows that are BSE resistant, and he has a herd of
cows.
yeah, but these are cloned cows, and he's also got a herd of cloned pigs where they've humanized them. They've removed this cell surface marker that stimulates an immune response, and he's cloned the dog. And as Alan Trounston said, he thought the human lines would turn out to be real and the dog would be a fake. Because the dog is very difficult. I mean, these guys are, you walk into the lab and I mean, it's obvious that they're really good. I'm still reserving judgment because I don't think he could have manipulated all of this himself, nor do I think he could have gotten 35 or 40 people in his research team to all buy into this. Let's do this. And why would you be so stupid after you've gotten away with the first paper where you've clearly staked your name to come back with something even more audacious that at some level you would have to back up? I mean, he was talking about exporting his technology to the UK and to the US with his people going out and do this. And yet they can only do it in Korea. I mean, at some point he would have been found out. So I'm very confused about all of this.
But everyone was taken, even the best in the country.
Yeah, no, I agree. That's why I still want to see who did what, and this investigation that the Korean government's undertaking, I think, will get at the very bottom of who did what, and then we'll have some, but the fact that they got hundreds of blastocysts following somatic cell nuclear transfer suggests that they were very good at what they were doing. I think there probably were cell lines, and where are they?
Thank you, Steve. I have to tell you that unfortunately, Bill, I've been involved with a number of examples of scientific misconduct in which people in collaborative studies have published in the open literature in violation of the rules of the study and didn't understand that they were, or couldn't seem to understand they were going to be discovered just by virtue of the publication. I mean, so it defies logic to some extent. Our second speaker is Dr. Karen Hershey, who's already been referred to by Dr. Minger. Dr. Hershey's from the Baylor College of Medicine, where she's an associate professor in the departments of pediatrics and molecular and cellular biology in the Center for Cell and Gene Therapy at Baylor. She's also part of the program in developmental biology of the Baylor College of Medicine and an adjunct assistant professor in the Department of Bioengineering at Rice University. Again, multidisciplinary, multi-campus. One of the foci of her laboratory is to investigate the potential of adult and embryonic stem and progenitor cells to contribute to neovascularization in response to tissue injury and growth. She graduated with a BS from Penn State, her PhD from the University of Arizona, and today she'll be reviewing some of her research as well as other works being done in the Stem Cell and Regenerative Medicine Center, or the STAR Center at Baylor, where she's the deputy director. Karen?
Thank you very much, Ken. And I'd like to thank all of the organizers for the opportunity to come over and tell you about the newly formed STAR Center at Baylor. And how do we advance? Okay. So I'd like to give you an overview of the STAR Center, who we are and the research that we're doing, and also to give you some idea about the collaborative research that we have ongoing with different institutions within the Texas Medical Center, and how our efforts trying to bring together the study of basic biology with that of bioimaging and bioengineering so that we can truly make advances in the area of regenerative medicine. And then I'll also tell you about some work that my lab is doing, trying to understand how blood vessels form in the embryo and how this feeds into the concept of vascular regeneration for adult tissues, and then how this works with other investigators for the promotion of tissue engineering and tissue regeneration on the whole. So the STAR Center really emerged from within the Center for Cell and Gene Therapy at Baylor, which is directed by Malcolm Brenner. And he established this center about 8 years ago, and it really has a broad mission for promotion of gene therapy and vector development, as well as for the development of cell therapies. And there was a group of people, a nucleus of people, really with a more focused interest in stem cell biology and using this technology for tissue regeneration. And this group came together to form this STAR Center, STAR standing for Stem Cell and Regenerative Medicine. And so we're really sort of a sub-center of the Center for Cell and Gene Therapy, which is advantageous to us because we're still under that administrative umbrella. And it gives us some flexibility in developing our own infrastructure while our bills are paid by the CAGT. So that's really nice. It's like shopping with your dad's credit card. So we also have additional support, financial and developmental support from the Baylor administration. And we're financially supported as well by individual grants that the investigators within the center have gotten from various federal agencies, including the NIH. And we've now started to get private donations, which we're very grateful for, and will help to establish our infrastructure and to propel our research. So the mission of our center is to facilitate stem cell research of all types at Baylor and to enable the clinical translation of stem cell research for regenerative medicine. This keyboard doesn't like me. Makes me do it twice. So the investigators within the center currently, our director is Margaret or Peggy Goodell, who is a basic science researcher who studies hematopoietic stem cells and is very interested in the characterization of these cells and understanding how their growth and their differentiation is regulated. So really a very basic stem cell biologist. Myself as the deputy director, and my lab studies the process of blood vessel formation during development with a strong interest in vascular regeneration in adult tissues, and I'll tell you a little bit more about our work specifically in a little bit. And then Thomas Waka is the first hire into the STAR Center, and he did his postdoctoral work with Jamie Thompson, the lab in which the first human embryonic stem cells lines were derived. And Thomas was the person within Jamie's lab to pioneer genetic manipulation of human embryonic stem cells for homologous recombination. And we have other members of the center, so these are the primary members within the center, and then we have other associates that are also members of the CAGT, and they're listed here, and they have various expertise in stem cell biology and regeneration of various tissues such as bone and neural regeneration, and then other enabling technologies such as vector development, which is an interest of Mike Berry and Richard Sutton. And then there are other associate members of the STAR Center throughout Baylor campus, and some of them are listed here, and this is not comprehensive. I think there are about 40 members total within our STAR Center. And there are various expertise, including our own Bill Brinkley, who will do the closing remarks, who's very interested in mammary gland stem cells as well as really invested in stem cell policy. We have other investigators studying embryonic stem cells, and doing bioimaging, also an enabling technology that's required for tissue regeneration, and then other investigators, both clinical, translational, and as well as basic scientists. And please check out our website with a complete listing of all of the investigators and their expertise. So the goals of our group collectively are to stimulate and train investigators at Baylor to work with stem cells, particularly providing training in the use of human embryonic stem cells, now with Thomas's expertise. We would like to build research infrastructure within our center to support training and ongoing research in this field, and then also to recruit in new junior investigators with a focus in stem cell research with an eye toward regenerative medicine. And we currently have an ongoing search for 3 junior investigators, so hopefully we'll be adding to our team shortly. We also have an interest in expanding our interactions within the medical center more broadly. We already have significant interactions and collaborations ongoing with Rice University, and I'll tell you a little bit more about those specific projects. And we're hoping to develop closer ties with other institutions that are also developing stem cell research programs and programs in regenerative medicine. And so it's our hope that as we're all building these programs that we could collect resources and have more of a synergy enabling and empowering Texas Medical Center and the resources that we have to give us a better power position within the U.S. In terms of tissue regeneration, stem cell technology. And so I want to say a little bit more about the collaborations that we have established with Baylor and about the programs that we have ongoing. And so in these projects, we're really trying to bring together the expertise of both of these institutions. So within the CAGT, and particularly within STAR, we have a good foundation in basic stem cell biology, and also a good understanding of developmental biology and good enabling technologies such as bioimaging. And we're trying to merge this with expertise in the bioengineering department at Rice, which includes development and adaptation of biomaterials, as well as bioreactors and nanotechnology. And so we're bringing all of these resources together, pooling expertise and resources to try and do some projects in the area of tissue engineering for regenerative medicine. And some of the projects that we have ongoing are listed here. And this is one, a bioengineering research partnership that was recently funded by NIH, the National Institute for Bioimaging and Bioengineering. And this project is on the tissue engineering of hematopoietic bone. And it involves multiple investigators within our STAR Center, and they're listed here, as well as investigators at Rice, both Jennifer West and Tony Mikos. And many of us within the STAR Center also have joint appointments at Rice as well. Sorry. So another project that we have in the works, which I think is also very exciting and will be very beneficial for regenerative medicine, is a quantum grant project, which hopefully will be funded also by the NIBIB. And this is a project that has a big question mark because it's actually being submitted today. And I have to apologize for having to leave shortly after my talk because we're having some problems printing and it has to be FedEx this afternoon. And so, but if all goes well, I'll be back for the reception. And so this project is really very exciting. And it's focused on the regeneration of both the neural system and the vascular system for the repair of prevalent disorders such as stroke and Huntington's and Parkinson's disease as well. And again, it's a very big collaborative effort and involves people from Baylor and Rice. Oh yeah, thanks. From investigators at Baylor and Rice, including myself as the PI, Mary Dickinson, who's a bioimager, Jennifer West, who develops biomaterials and bioreactors for tissue regeneration, Malcolm Brenner, the CAGT director, who studies hematopoietic stem cells and mesenchymal stem cells from human marrow with an effort toward generating neural stem cells from those cell sources. And then Thomas, of course, who works on human embryonic stem cells, using these again as sources for neuronal and vascular progenitor cells for these tissue engineering projects. And this particular project is also a collaborative effort with colleagues in the UK. Robin Lovell-Badge is the co-PI, and he's at the National Institute for Medical Research in London. And Jack Price and Mike Moto also are working with us in there at King's College, where Stephen is. So I also want to tell you a little bit more about this bioengineering research partnership to give you an idea of how investigators are really pooling their efforts and their expertise to work on a particular problem. And this is just hopefully one of many projects like this that we'll have. And so the charge of this project really is to engineer hematopoietic bone, which looks fairly complex. And this is a cross-section, a schematic cross-section of a long bone, just to show you what the structure looks like. And this is cortical bone, and within it is a marrow cavity. And in a long bone, there is a central artery that feeds the bone and feeds the marrow and out into the cortical bone structure. And so what our project is to do is to recapitulate this bone structure in vitro in a bioreactor system that was created, two bioreactor systems, one created by Tony Mikos to form bone tissue, and one created by Jennifer West to form vascular grafts. And so the idea is to make a vessel graft, to build a bone marrow around it, and encase it in cortical bone. And if this is successful, then we will hopefully be creating an environment, a marrow environment, in which we can propagate hematopoietic stem cells ex vivo, which is something that hasn't been done very well at all to date. And then hopefully also generating a viable, meaning perfused and innervated, a transplantable bone that could be used for mending critical size defects, bone defects. And so this is a big undertaking, and when we looked at this project and after we got it funded, we were saying, how are we going to make a hematopoietic bone? But luckily we said that after we got the money to do it. And so you can see the list of investigators involved in this project, and this is really very complex and requires all of these investigators wrapping their mind around this project and really understanding how their component feeds into it, and then having organizational meetings where we can all talk about how are we going to put this together and come out with the same endpoint, the hematopoietic bone. And so my role is not only in the administrative gathering of all the groups and organizing their efforts, but also trying to understand how bone is vascularized and how are we going to recapitulate blood vessel formation ex vivo. Alan and Betsy Davis provide expertise in understanding bone development. What is the process of bone formation? And if we're going to do this ex vivo, we really should know a lot about it. So they study this in vivo in development and also heterotopically in muscle tissue. They also have expertise in vectorology, which is very helpful if we have to transduce cells with particular genes to get the process going or to control it in another way. Peggy Goodell is also involved as our hematopoietic stem cell expert, trying to understand what is the environment in which hematopoietic stem cells reside and what needs to be recapitulated ex vivo so that the cells, their growth and the differentiation potential can be controlled. Dave Shine, another investigator within our group, works on innervation, studying neural regeneration in response to various injuries, and he'll be contributing to the innervation of this engineered bone. And then we have imagers such as Mike Barry and Eva Sevic-Maraca, and they'll be helping us to track the cells within the implant. After we've implanted it, we need to know where are the cells going and how are they behaving. And so they'll be assisting with that as well. And Mary Dickinson is another imager who is also involved, but really from the other end, from the developmental standpoint. Her group is working together with mine to try and understand how marrow is established and how is it vascularized. So watching this process in real time during embryonic development, which is Mary's forte, is really going to be helpful when we think about recapitulating this process. And of course, we have Thomas involved as well as providing a source for the cells that will make this tissue engineered graft. And then the expertise from bioengineering at Rice includes Jennifer's capacity to do vascular tissue engineering, to provide biomaterials and bioreactors for the generation of tissues ex vivo, and nanotechnology as we need it. And then Tony has a lot of experience in bone tissue engineering, also in the development of biomaterials and bioreactors that will be used for various components of this project. And so from my perspective, I just want to tell you about the research that we're doing and how it feeds into these kinds of projects, and how everyone's collective expertise is really needed to move projects like this forward. And so when we look at this project, we say, oh gosh, what do we need to be able to do it, really? Well, we need to understand the steps that are involved in the formation of blood vessel structures. So how is a blood vessel normally formed endogenously? And then what are the molecular signaling controls for each step of this process? And then once we know that information, how are we going to be able to direct the process ex vivo? So outside of the body, we don't have the normal blood flow properties, we don't have normal oxygenation, and these are all things that we must consider when we're trying to design bioreactor systems to mimic what's happening in the body. And then, of course, we need cell sources for generating blood vessels, as well as other tissues, as Stephen had talked about. And so really what my lab does is studies vascular development. We try to understand how cell growth and differentiation are controlled when vessels are forming. And then this feeds into the other applications, as I talked about, the engineering of vessels ex vivo, but also the manipulation of vessel formation endogenously for tissue repair, for vascularization of ischemic tissues, such as cardiac tissues or peripheral tissues. So this is a schematic just showing you the individual steps involved in the formation of a blood vessel. So this process alone is quite complex, and the molecular signaling required for each of these steps is different. And so the various parts of vessel formation that we're interested in are listed here. And of course, this is not comprehensive. There's many things that are missing off this list that we don't study, such as the formation of a lumen, also the specification of endothelial cells to arterial or venous endothelial cells. These are also very important when you're talking about making a vascular network. But these are the kinds of things that my lab studies, excuse me, which is the differentiation of primitive mesodermal cells that are multipotent into endothelial cells. So how does a cell decide to commit specifically to a vascular endothelial cell lineage? What are the molecular signals that are needed to drive this in vitro? And then once endothelial cells are formed, how are they specified? And we're particularly interested in specification to hemogenic endothelial cells, those that have the capacity to give rise to all blood cells. And then once a primitive vasculature is formed, it has to be remodeled into a circulatory network. And this requires very coordinated regulation of endothelial cell proliferation as well as migration, which I'll talk a little bit more about. And then vessel wall assembly. Once an endothelial cell tube is formed, it needs to acquire contractile cells around it for the control of blood flow. And these are smooth muscle cells or pericytes that are recruited by the endothelial cells themselves. And so these are the kinds of signaling processes that we study. And we do all of our studies in the mouse embryo model system. And a little mouse embryo is shown here at about day 8 of gestation, and the normal gestation for mice is 21 days. So at this stage of development, the mass becomes so large that it needs to induce formation of its own vascular structures for continued perfusion, oxygen perfusion, and distribution of nutrients and removal of waste products. And vessels form in the mouse embryo first in a tissue that's actually surrounding the embryo. This is the embryo proper inside. And this tissue surrounding it is the yolk sac. And this is the first site of vessel formation as well as hematopoiesis in most mammals, including humans. And interestingly, when vessels are forming, they really don't form into a circulatory network initially. They form into this net-like structure referred to as a vascular plexus. And then this has to be remodeled into a circulatory network. And we like to study vessel formation in a mouse in this yolk sac because, well, first of all, the mouse is genetically manipulatable. So we're interested in signaling pathways and how various pathways converge to form structures in morphogenesis. And so we can manipulate a single pathway and understand what is the impact of that manipulation on the whole process. Also, this is monitorable in vivo. We can watch using bioimaging techniques how vessels are forming and watch migration and proliferation and differentiation of the cell types in real time. So for those reasons, we like this model system and we like to study vessel formation in this yolk sac tissue. And here's a cross-section of the tissue just to show you the simple structure, which is really just 2 layers of visceral endoderm and a mesoderm. And by doing molecular genetic manipulation of the mice, we can determine what is the signaling pathway that regulates commitment of mesodermal cells to an endothelial cell fate. And so this is just an overview of those signaling pathways, and the specific molecules that are involved are really not important for this purpose, but more important is that we can dissect the signaling pathway and understand how mesodermal cells that are really multipotent, and they have choices about what cell type they can form, and how are they directed to be specifically primitive endothelium. And the idea is then we would take these factors exogenously and provide them to mesodermal progenitors in vitro and able to form a vascular plexus of primitive endothelium. Of course, cell source is important. We're using mesodermal progenitors, but provided from where? And this is something I'll come back to at the end. Of course, then once primitive endothelium is formed, it has to be further specified to venous or arterial or hemogenic endothelium, and this involves not only soluble signals and coordination of molecular pathways, but also physical forces, plasma flow in the embryo, and how is this mimicked ex vivo so that we can really generate arterial endothelial cells or venous endothelial cells for tissue engineering projects. And then, of course, once you have a plexus, it has to be remodeled into a beautiful circulatory network. And this requires the coordination of cell proliferation and migration. And again, using genetic tools, we've been able to dissect the pathways that regulate these processes to form vessel structures. And so we know that this is very complex because in the mouse system, there are about 50 different mutants that have the same phenotype. They develop to some point in development, and they arrest then because they form a plexus that never becomes remodeled into a circulatory network. So using these embryos in a culture system, where we took the mouse embryos, we dissected them out at day 7.5 when vessels were just forming, and we manipulated them in culture, adding back exogenous factors that they were lacking in the mutants. And then to summarize 3 years worth of work in one slide, what we found was that interestingly, when endothelial cells are forming vessel structures and this is being remodeled, the migration of the cells and the growth of the cells is independently regulated by different signaling pathways. We actually didn't know that before, and now that we know it, we're even more frightened by having to recapitulate that in vitro. And so what we know that there are various signals, the go signals that tell the cell to proliferate, and then there are other signals that tell the cell to stop proliferating. And the matrix that's made by these cells is very instrumental in survival of the cells and mediating migration and proliferation. So we've learned quite a lot from these embryonic studies that we think will be useful for directing this process in vitro. Okay, so how are we going to apply this to these tissue engineering projects? The things that we're involved in, particularly with this bioengineering research partnership, are the fabrication of the arterial graft that will be in the center of the bone, the vascularization of the marrow, enabling vascularization from a central artery into the other parts of the bone. And then what is the role of the vascular cells in supporting the marrow niche and their association with hematopoietic stem cells? How does this control their growth and their differentiation? And then what about interdependence of vascularization and innervation, which seems to occur in parallel in the embryo and in the adult? And then, of course, identification of cells that we're going to use for vascular tissue engineering. And in this regard, it's actually a very important question. When patients need vascular grafts or vascular regeneration, the vascular cells that are currently present as mature cells within their vessels really don't behave well in culture. They don't grow very well, so we have to think about other human cell sources. And so we're currently trying to identify and isolate progenitor cells from adult tissues that would be used as autologous cells, and those are included cells from bone marrow, blood, and skeletal muscle. But because these are very limited, as Stephen had mentioned earlier, we're also trying to harness the potential of the human embryonic stem cell system to derive vascular cell types. And so in my lab, we established the human embryonic stem cell system with Stephen's help and a lot of email and phone conversations. And we are currently creating reporter cell lines with Thomas's lab so that we can mark the differentiation of these cells. And then we'll use the information that we have from our developmental systems and molecular regulation to drive the cells to differentiate them toward endothelium and smooth muscle cells for vascular engineering. So I think that's all. I just want to thank all the people in my lab and all my collaborators for their hard work on these projects.
Thank you very much. Questions? for Dr. Hershey? Yes, please.
As a former member of NIBIB from the administrative side, one of the goals is very much to integrate biologists with bioengineers. And in this project, you have a BRP as well as potentially a quantum grant. I was wondering, what was the role of the bioengineers
The role of bioengineers
in this project? Yeah.
Well, they can probably tell you better than I, not understanding their bioreactor systems. But so their goals are actually to provide the things that they create. They create biomaterials. They adapt them for the adhesion of cells. They have to be adaptable to be degradable in a certain time frame in vivo. So they provide all of that support. We provide the biology, we provide the cell types, but they really have to provide the scaffolds, the bioreactors, the nanotechnology as it's needed for cell tracking. So they provide quite a lot to our team.
Have they started providing it? Because I see
Yes.
the biological systems.
Yeah, you see the biological systems because I presented the talk. So, for us, there are enabling technology, but for them, we're their enabling technology. So it would be quite different if Jennifer was up here giving this talk, and I'd be like, oh yeah, well, we got the cells from her, and So it's a different perspective, and that's why you maybe have read it out that way. But they're just as important as we are.
Other questions? Maybe I missed it at the end when you were talking about embryonic cells, but knowing what you know about growth factors, is there any data that indicates that you could induce vascular formation with stem cells using the pathways that you've identified?
Yeah, definitely. So the pathways that we've identified in the mouse embryonic stem cell system, we've been able to apply that to human embryonic stem cells, not in a very efficient, not reliably as much as I would want, but yes, we can use the same pathways to drive commitment of human embryonic stem cells.
And how far down the developmental line would those stem cells go? I mean,
we're so early in the studies that I wouldn't want to say.
Well, maybe next time you'll tell us that
Yeah.
you can form blood vessels. Thank you very much.
Okay, thank you.